Hello everyone, myself Amrita Yadav, Assistant Professor, School of Pharmacy, BBTU. Today I am going to discuss about electrophoresis. Introduction of electrophoresis. Definition of electrophoresis. Electrophoresis refers to the movement of charged particles and macromolecular ions under the influence of an electric field. Depending on the sign of their net charges, these substances migrate either to the cathode or to the anode. The differences in migration velocities, which provide a powerful means for the analysis and separation of substances which are difficult to fractionate by other methods. Now, electrophoretic mobility. The migration velocity of a particle when the electric field acting on it is 1 volt per centimeter is known as electrophoretic mobility. Electrophoresis is a simple, rapid and highly sensitive method. It is used for the separation of body proteins in body fluids such as serum, urine, CSF cerebrospinal fluid, protein in erythrocytes, example hemoglobin, nucleic acids, DNA or RNA. Principle of electrophoresis. Any charged ion or molecule migrates when placed in an electric field. An electrophoretic system consists of two electrodes of opposite charge, anode and cathode, connected by a conducting medium called an electrolyte. The separation effect on the ionic particles result from differences in their velocity, that is nu, which is the product of the particles' mobility and the field strength. Nu is equal to m into E. The rate of migration depends upon number of factors, net charge of molecule, size and shape of the particles, strength of electrical field, properties of supporting medium and temperature of the operation. This will be discussed later on. Clinical application of electrophoresis, nucleic acid analysis. It is the identification and study of DNA and DNA fragments. DNA is notable for the consistency of its negative charge, which means that the electric current applies roughly equal forces to any portion of DNA. Under that pressure, larger and smaller fragments of DNA begins to separate because they all are affected differently by frictions from the test medium. The medium, usually arrows gel or an acrylamide gel, freezes the separated segments in place when the current is removed, which allows them to be examined in high resolution. Staining agents such as ethium bromide are often added to the gel to make it easier to see and interpret the result. Second, protein and antibiotic interactions. Another common form of electrophoresis is immunoelectrophoresis, which analyzes the presence and behavior of certain proteins. Many medical conditions, including multiple sclerosis, kidney disease, and some cancer, result in the creation of abnormal protein molecules. These molecules can be detected by performing electrophoresis on urine or blood samples and watching for any variance from the normal quantities and types of protein. Immunoelectrophoresis can also be used to detect specific proteins called immunoglobulins, which act as antibodies. These are part of the body's immune system and attack foreign proteins such as viruses and allergens. Analyzing these antibodies can be helpful to identify new therapies to treat those invaders and also provides insight into conditions such as allergies and autoimmune disorders, which can result from malfunctioning antibodies. Third, antibiotic testing of antibiotics. Electrophoresis plays a number of roles in the testing of antibiotics. One of the most common is testing the purity of an antibiotic. This can be done by applying electrophoresis to a solution which contains antibiotic in the form of a paper strip impregnated with the antibiotic or a capillary filled with the solution. One can differentiate between the antibiotic itself and any impurities present along with it. They can also determine how concentrated the antibiotic is, which is crucial for applying accurate dosage form. Another application is testing vaccines. Electrophoresis is useful in both the creation and production of vaccines. The purpose of a vaccine is to help the body generate antibiotics to a potentially dangerous pathogens, and electrophoresis is useful method for detecting those antibiotics. It can be used to compare the effect of a vaccine or multiple versions of a vaccine across a large number of test subjects or other variables. Once the vaccine is in production, electrophoresis also provides a quick and effective way of testing production batch-wise. It can also be used in serum protein electrophoresis, lipoprotein analysis, diagnosis of hemoglobinopathies and hemoglobin A1C. Hemoglobinopathies these are inherited single gene disorders. In most cases, they are inherited as autosomal codominant traits, for example, sickle cell diseases. Hemoglobin A1C, these are glycosylated hemoglobin. It is one of the hemoglobin that is chemically linked to a sugar. Determination of serum protein phenotypes and microheterogeneities. For example, alpha-1 antitrypsin deficiency or MM. MM is a meningomylosis. It is a sign which are concerned with embryology or pediatric. It is used to study the congenital defects. Genotyping of proteins, for example, EPO analysis of Alzheimer's disease. EPO analysis is a study which involves EPO-E and EPO-CL, which are associated with the dementia in Alzheimer's disease. Factors affecting electrophoresis. This include internal factors and external factors. Internal factors include magnitude of its charge, charge density, molecular weight, shape. External factors include pH of the solution, electric field, viscosity of the solution and temperature. Charge, size and shape of the sample being separated affects its own migration rate. A net increase in the charge increases the rate of migration. In accordance with the Henderson-Hazelbach equation, magnitude of charge is pH dependent. Rate of migration is affected by increase in size of molecule, which has inversely proportional relationship, and difference in shape of the sample. Therefore, larger charge will move faster, whereas larger mass moves slowly because the frictional forces slow down the molecule traveling through the solution, which depends on molecular size. Rate of migration increases with increasing net charge. It depends on pH of the solution. Size. Rate of migration decreases for larger molecules due to increase in frictional and electrostatic forces. Shape. Molecular have similar size but differ in shape, exhibit different migration rate. External factors. Electric field. An electric field is a property that describes the space that surrounds electrically charged particle. It is defined as the electric force per unit charge. This electric field exerts a force on other charged object and is radially outward from a positive charge and radially inwards a negative charge. A movement of ions depends upon voltage, current and resistance of the electric field. Viscosity of the solution. Viscosity of the solution. As the viscosity of the medium increases, the rate of migration of charged ions decreases and vice versa. Temperature. Temperature affects mobility indirectly since viscosity of the supporting medium is temperature dependent. pH. The extent of ionization depends on pH. The ionization increases with increase in pH for organic compounds, affecting its rate of migration, whereas the ionization decreases with increase in pH for organic bases. Types of electrophoresis. It is divided into two: zone electrophoresis and moving boundary electrophoresis. Zone electrophoresis consists of paper electrophoresis, gel electrophoresis, thin layer electrophoresis, and lastly cellulose acetate electrophoresis. Now coming to the moving boundary electrophoresis, it consists of capillary electrophoresis, isotachophoresis, isoelectric focusing, and lastly immunoelectrophoresis. Paper electrophoresis. Paper electrophoresis is the form of electrophoresis that is carried out on filter paper. This technique is useful for the separation of small charged molecules such as amino acids, small proteins. In this, filter paper can be used as a stabilizing medium. For example, Whatman filter paper, cellulose acetate filter paper, or chromatography paper. Apparatus used for this, it consists of power packed electrophoretic cell that contains electrodes, buffer reservoir, and finally support for paper. Gel electrophoresis. Before starting gel electrophoresis, first we discuss about what is gel. What is gel? Gel is a cross-linked polymer whose composition and porosity is chosen based on the specific weight and porosity of the target molecules. Gel electrophoresis, it is a technique used for the separation of DNA or RNA or protein molecules according to their size and electrical charge using an electric current applied to a gel matrix. Types of gel. There are two types of gels which are used in gel electrophoresis: agarose gel and polyacrylamide gel. Agarose gel electrophoresis, it is highly purified uncharged polysaccharide derived from agar. It is used to separate macromolecules such as nucleic acid, large proteins, protein complexes, and it is prepared by dissolving 0.5% agarose in boiling water and allowing it to cool to 40 degrees centigrade. It is a fragile because of the formation of weak hydrogen bonds and hydrophobic bonds. Next is polyacrylamide gel electrophoresis. It is frequently referred to as PAGE, that is polyacrylamide gel electrophoresis. Cross-linked polyamide gel are formed from the polymerization of the monomer in the presence of small amount of N-methylene bisacrylamide. Bisacrylamide, it is two acrylamide linked by the methylene group. The polymerization of acrylamide is an example for free radical catalysis. They are defined in terms of total percentage of acrylamide present and pore size vary with the concentration. Commonly used components are acrylamide monomers, ammonium persulfate, tetramethylene diamine, and N-methylene bisacrylamide. These are free radical activate acrylamide monomers, inducing them to react with other acrylamide monomers, forming long chains. It is used to separate most proteins and small oligonucleotides because of the presence of small pores. Now, cellulose acetic electrophoresis. Thermoplastin resin made by treating cellulose with acetic anhydride to acetylate the hydroxyl group. When dry, membrane contains about 80% air space within fibers and brittle film. As the film is soaked in buffer, the space are filled. Because of their opacity, the film has to be made transparent by soaking in 95:5 methanol glacial acetic acid. It can be stored for longer duration. Capillary electrophoresis. It is an analytical technique that used ions based on their electrophoretic mobility with the use of an applied voltage. The electrophoretic mobility is dependent upon the number of factors. Number one, the charge of the molecule, the viscosity, and the atom's radius. The rate at which the particle moves is directly proportional to the applied electric field. The greater the field strength, the faster is the mobility. Neutral species are not affected, only ions moves with the electric field. If two ions are having the same size, the one with greater charge will move the fastest. For ions of the same charge, the smaller particle has less friction and overall faster migration rate. Capillary electrophoresis is used most predominantly because it gives faster result and provides high resolution separation. It is useful technique because there is a large range of detection methods are available. Types of capillary electrophoresis. There are 6 types of capillary electrophoresis. First, capillary zone electrophoresis. Second, capillary gel electrophoresis. Third, micellar electrokinetic capillary chromatography. Fourth, capillary electrochromatography. Fifth, capillary isoelectric focusing. And lastly, capillary isotechophoresis. Application of capillary electrophoresis. It is used for the simultaneous determination of the ions such as ammonium ion, potassium, sodium, magnesium and calcium ion in saliva. It is used in forensic science for the development of methods for application and detection of DNA fragments using polymerase chain reaction. It is cost-effective approach to do DNA sequencing. It is used for the intermolecular binding interactions to understand protein ligand interactions, and finally it is used in forensic biologist as typing of STR from biological samples to generate a profile from highly polymeric genetic markers which differ between individuals. Now we will discuss thin layer chromatography in detail. Today we are going to discuss about thin layer chromatography. Thin layer chromatography, which is also known as TLC. It is a technique which is used to separate component of a mixture. This thin layer chromatography can be performed on a sheet of glass, plastic or aluminum foil, which is coated with a thin layer of absorbent material. This absorbent material may be silica gel, aluminum oxide or cellulose. This adsorbent material, which is also known as the stationary phase, after the application of the sample on the plate, a solvent or a solvent mixture, which is known as mobile phase, is drawn up the plate through capillary action, because different analytes ascends the TLC plate at different rates and the separation is achieved. The silica, which is used as an adsorbent material in TLC plate, we can see this, that silica consists of the silicon, which is surrounded by the 3 molecules of oxygen and 1 molecule of hydroxyl ion. This is the main body of silica structure. Here we can see this is a solvent system or solvent chamber, which consists of a TLC plate on which a spot is applied, and this TLC plate is placed in a solvent chamber in which mobile phase travels upwards along with a spot. And here we can see after separation there are a number of spots which are separated, and these number of separated spots are component of a mixture. Principle of TLC. In chromatography, 2 types of principles are involved, namely partition and adsorption. Here we will discuss both type of principles. First one is partition. The separation of the components from the sample mixture is carried out by the process of partition of the component between 2 phases. If both phases are in liquid form, in this process the immiscible solid surface coated with the liquid surface on the stationary phase is in the mobile phase. The liquid surface is immobilized by a stationary phase, which result in making it a stationary phase. The mobile phase moves from the stationary phase and the components gets separated. The separation totally depends on different partition coefficient. Types of partition chromatography: liquid chromatography, gas liquid chromatography. Now coming to the adsorption. Adsorption chromatography. In this chromatography, the mobile phase may be a liquid or a gas. The stationary phase is always a solid, and the separation is based on the principle of adsorption. The sample, the compounds that shows more affinity with the stationary phase adsorbs on the surface of the stationary phase and retain maximum time on this stationary phase on the basis of the affinity of the compound with the stationary phase, and the compounds that shows less affinity with the stationary phase are elute out very easily with the mobile phase. Adsorption chromatography. which involves the analytical separation of a chemical mixture which is based on the interaction of the adsorbate with the adsorbent. The mixture of a gas or liquid gets separated when it passes over the adsorbent bed that absorbs different compounds at different rates. Now, what is adsorbent? Adsorbent, a substance which is generally porous in nature with high surface area to adsorb substances on its surface by intermolecular forces, is known as adsorbent. For example, silica gel H, silica gel G, silica gel N, silica gel S, hydrated silica gel, cellulose microcrystalline, alumini, or modified silica gel, etc. RF values, the retention factor or RF is defined as the distance travelled by the compound divided by the distance travelled by the solvent. For example, if a compound travels 2.1 cm and the solvent front travels 2.8 cm, then the RF value will be 0.75. The RF value for a compound is a constant from one experiment to the next only if the chromatographic conditions are constant. What are the chromatographic conditions? These are solvent system, adsorbent, thickness of the adsorbent, amount of the material spotted, and finally the temperature. Since these factors are difficult to keep constant from experiment to experiment, therefore here we can use relative RF values which are generally considered. Relative RF means that the values are reported relative to a standard, or it means that you compare the RF values of one compound run on the same plate at the same time. The larger RF of a compound, the larger the distance it travels on the PLC plate. When comparing two different compounds run under identical chromatographic conditions, the compound with larger RF is less polar because it interacts less strongly with the polar adsorbent on the TLC plate. Vice versa, if you know that the structure of the compound in a mixture, you can predict that a compound of low polarity will have larger RF values than a polar compound run on the same plate. The RF can provide corroborative evidence as to the identity of a compound. If identity of a compound is suspected but not yet proven, an authentic sample of the compound or a standard is spotted and run on a TLC plate side by side or on top of each other with the compound in question. If two substances have the same RF values, they are likely the same compound. If they have different RF values, they are definitely different compounds. The RF value is constant for each component only under identical experimental conditions, and it depends upon the number of factors, namely nature of adsorbent, the mobile phase, temperature, developing tank, mass of the sample applied, chromatographic techniques, etc. Now we will discuss all these factors in details. First, nature of adsorbent. Different adsorbent will give different RF values for the same solvent. Reproducibility is only possible for given adsorbent of constant particle size and binder. Plates should be stored over silica gel in desiccators before use, and the sample should be applied quickly so that the water vapor in the atmosphere is not absorbed by the plate. Because of the difficulties associated with the activation procedure, it is far better to use plate stored at room temperature and not to activate them. Next one is the mobile phase. The purity of solvent and the quantity of solvent mixed should be strictly controlled. It should be made freshly for each run if one of the solvent is very volatile or hygroscopic, for example acetone. Now coming to the temperature, although precise control of temperature is very necessary, That the tank should be kept away from the source of heat, direct sunlight, etc. As the temperature is increased, volatile solvents evaporate more quickly. Solvents run faster and RF values generally decreases slightly. Thickness of layer. Standard plates approximately is of 250 micrometer is the preferable thickness of the layer. Below 200 the RF values vary considerably. The layer may be of higher to lower thickness in individual compounds. Developing tank, it is important that saturated conditions should be attained for running TLC plates. This is the best accomplished by using small tanks with filter paper liners and sufficient solvents, and by leaving the tank to equilibrate for at least 30 minutes before running the plates. Mass of the sample. Increasing the mass of sample on the plate will often increases the RF of the drug, especially if it is normally tails in the system. However, if a plate is grossly overloaded, this too will give a tailing spot and will have the effect of apparently decreasing the RF values. The two situations are normally easy to distinguish between the intensity of the spot. Last one is the chromatographic technique. Depending upon the development technique, namely ascending, descending, horizontal, etc., the RF value changes for the same solvent system. How to run a TLC plate. First, prepare the developing container. In this, the developing container for TLC can be specially designed chamber, a jar with a lid, or a beaker with a watch glass on the top. Pour solvent into the chamber to a depth of just less than 0.5 centimeter. To help in the saturation of the TLC chamber with solvent vapors, you can line part of the inside of the beaker with filter paper. Now cover the beaker with a watch glass and allow it to stand while you are preparing TLC plate. Now coming to the TLC plate preparation. In TLC plate preparation, we can make a slurry of suitable adsorbent materials such as silica gel, alumina, etc. with a small amount of inert binder like calcium sulfate, which is also known as gypsum, with water. This mixture is spread as a thick slurry on an unreactive carrier sheet, usually alumina, foil, glass, plastic, etc. Now, after pouring the slurry on the plate, it can be air dried and finally it can be heated in an oven for 30 minutes at 110 degrees centigrade. This process is known as activation of TLC plate. Now, what is activation of TLC plate? Usually the TLC plate in contact with atmosphere absorbs some amount of moisture. In order to remove that amount of moisture, it is activated in an oven for 30 minutes at 110 degrees centigrade. Keeping in this mind, the thickness of the adsorbent layer is or should be 0.1 to 0.25 mm in case of analytical purpose and around 0.5 to 2 mm for preparative TLC. Next is spotting the TLC plate. If the sample is not ready in solution, dissolve about 1 mg in 1 ml of volatile solvents such as hexanes, ethyl acetate or methylene chloride. As a rule of thumb, a concentration of 1% usually works well for TLC analysis. If the sample is too concentrated, it will run as a smear or streak. You will see nothing on the plate. Sometimes you will need to use trial and error to get well-sized, easy to read spots. Now, how to apply the solution on the TLC plate? It can be done by a microcapillary For example, in the organic labs, we use 10 microliter microcaps. They are easier to handle than the smaller ones used in other labs. Dip the microcap into the solution and then gently touch on the at the end of the TLC plate with location on the TLC plate. Don't allow pot to become too large, otherwise it will create a troublesome in resolution of the spots. Spotting of the plate. The thin end of the spotter is placed in the dilute solution. The solution will rise up in the capillary, that is the spotting or the spotter. This spotter is touched on the plate at the starting line and the solvent is allowed to evaporate along with the spot at the same place again. This way you will get a concentrated and small spot. Try to avoid spotting too much material because this will deteriorate the quality of the separation considerably, which is result in tailing. The spots should be far enough from the edges and from each other as well. If it is possible, you should spot the compound or mixture together with the starting material and the possible intermediate on the plates. This can help you in getting the separated or in comparison the reaction of the mixtures. Now, how the development of the solvent is done. The choice of a suitable solvent is totally depend upon the nature of the substance and the adsorbent which is used during the chromatography. A development solvent should be such that it does not react chemically with the substances in the mixture which are under examination. For example, carcinogenic solvents, for example benzene, etc., or environmentally dangerous solvents, example dichloromethane, should always be avoided. Solvent system range from non-polar to polar solvents. Non-polar solvents are generally used as highly polar solvents cause the adsorption of any component of the solvent mixture. The commonly used development solvents are petroleum ether, carbon tetrachloride, pyridine, glycol, glycerol, diethyl ether, formamide, methanol, ethanol, acetone, and N-propanol. After that, visualization of the spot is done when the solvent front has moved to about 1 centimeter of the top end of the adsorbent. Approximately after 15 to 45 minutes, the plate should be removed from the developing chamber and the position of the solvent front marked with a pencil, and the solvent is allowed to evaporate. If the component of the sample are colored, they can be observed directly. If it is not colored, they can be sometimes visualized by or with the help of ultraviolet light on the plate, or by allowing the plate to stand for few minutes in a closed container in which saturated iodine vapors are maintained. Sometimes the spots can be visualized by spraying the plate with a reagent that will react with one or more of the components of the sample, and it can indicate the color of the sample. Advantages of thin layer chromatography. Thin layer chromatography is very easy to separate the component of a mixture. It is a sensitive method. In comparison to other separation techniques, very few types of equipments are used in TLC. The components are separated in a very short time because the components will elute rapidly. It is feasible to visualize all components of UV light. The non-volatile compounds are separated by the TLC methods. Only small sample size is required in TLC and it can be microliter. A comparison with the starting material, tentative identification is possible. The component in the complex mixture of the samples are able to easily separate and recovered by stretching the plates. There are certain disadvantages of thin layer chromatography. There is no longer stationary are available in TLC plate. Therefore, its separation length is insufficient in comparison to other chromatographic techniques. The results which are obtained from TLC are difficult to reproduce. Only soluble components of the mixture are possible, and qualitative analysis is possible but not quantitative analysis. It is usually not automatic. TLC work in the open system, therefore due to temperature and humidity, it can affect the result also.